Hello, students. Welcome to today's high-yield revision session. Becco, we're not wasting any time today. No fluff, straight to the point. If you're preparing for your medical exams, you know how absolutely crucial the basics are, right? Today, we're doing a fast-paced, point-wise breakdown to lock these core biochemistry concepts into your memory forever. Farafat energy lao, set your focus, and let's dive right in. We are pulling today's core fundamentals directly from Harper's Illustrated Biochemistry 32nd edition. And listen, these aren't just your ordinary notes. We are analyzing this definitive text line by line. We're embedding the theory into clear points and flowcharts because, we aren't just smugging up, we're actually understanding the concepts from the ground up. Our syllabus for today covers 4 super important areas. First, water and hydrogen bonding. Second, hemoglobin and oxygen transport. Third, bioenergetics and ATP. And finally, cell cycle and DNA regulation. Chalo, let's start. Section 1. Water and hydrogen bonding, the true matrix of life. Let's start with tetrahedral geometry. A water molecule has a very specific 105-degree angle. Now, this isn't some random number to ignore. It's a guaranteed, crucial exam point, so much gay. This angled structure allows the water molecule to act as a dipole, dictating all of its unique interactive properties. Literally, without this exact 105-degree angle, life as we know it simply wouldn't exist. Water's hydrogen bonding dynamics are just fascinating. Water naturally self-associates via hydrogen bonds. But the real high-yield textbook highlight here is its dual role. A single water molecule serves simultaneously as both a hydrogen donor and a hydrogen acceptor. It can cluster with 4 neighboring molecules and strongly bond with other polar groups like alcohols and amines. What does that do? It drastically increases their solubility in our bodies. Egdom clear? Moving on, Section 2, hemoglobin and oxygen transport mechanisms. Listen up because this is a guaranteed long answer or multi-part MCQ topic for your exams. Pakka aayega. We are literally checking off the textbook's own chapter objectives for myoglobin and hemoglobin right here, ensuring not a single mark slips away. Structural similarities, binding curves, the Bohr effect, we're tackling the whole shebang right now. There's a sharp contrast between these two molecules. Both bind oxygen, but totally differently. Myoglobin has a hyperbolic binding curve, meaning it has a very high oxygen affinity, but it's honestly less efficient for bulk transport. Hemoglobin, on the other hand, has a sigmoidal or S-shaped curve. This magical curve comes from allosteric cooperative binding, giving it this superpower to load oxygen heavily in the lungs and unload it super easily in peripheral tissues. It is highly, highly efficient. Now, you need to lock these core concepts into your memory, okay? Distal histidine hinders carbon monoxide binding, which basically reduces CO toxicity. P50 is the partial pressure where hemoglobin is 50% saturated. This is a direct indicator of transport and delivery efficiency. Then you have proximal histidine, which pulls iron into the porphyrin ring upon oxygen binding, driving massive structural and conformational transitions. And don't forget 2,3-BPG. It's the allosteric regulator that promotes oxygen delivery to our tissues. This is pure clinical gold for your MCQs. Let's break down the Bohr effect into a logical, easy-to-remember sequence. Step 1. Red blood cells absorb CO2 from peripheral tissues. Step 2. This absorption causes structural changes in hemoglobin, dropping its oxygen affinity, which delivers the oxygen straight to the tissues. Step 3. This deoxygenated hemoglobin carries that CO2 back to the lungs. There, the oxygen-rich environment forces hemoglobin to bind oxygen again, successfully releasing the CO2 so we can simply exhale it out. A beautifully engineered system, right? Examiners absolutely love testing these clinical correlates. In sickle cell anemia, lowering the partial pressure of oxygen causes severe structural consequences to the mutated hemoglobin S. It literally polymerizes and sickles the cell. Meanwhile, thalassemias cause specific metabolic defects as a direct consequence of unequal production of alpha and beta chains, which leads to severe red blood cell destruction. Always, always link your basic science to the disease pathology. Section 3. Bioenergetics and ATP, the thermodynamics in biology. Dekho, don't let thermodynamics intimidate you. It's quite simple if we trace the flow. First law. Chemical energy from your food transforms into heat, electrical impulses, radiant or mechanical energy. Easy. Second law, for any spontaneous process to happen, the total system entropy must increase. Finally, the main equation links free energy change, delta G, and entropy change, delta S. Remember this, delta G must be negative for a spontaneous reaction.
This equation literally drives every single metabolic pathway in our body.
The absolutely crucial point here is entropy. Simply put, it's just the extent of disorder or randomness in a system. The textbook states clearly that as a system approaches equilibrium, entropy reaches its absolute maximum. Now, our cells are constantly battling against this equilibrium. They are spending energy in the form of ATP just to maintain order. So you see, life itself is a constant battle against entropy. All right, final stretch. Section 4, Cell Cycle and DNA. Strict regulation. Now let's see how a cell actually divides. Pay close attention to the prerequisites for the S and M phases. To divide, cells must assemble massive macromolecular complexes, like new chromosomes, and this is heavily dependent on specific molecular precursors. But most importantly, this entire process isn't random. It is strictly regulated by intricate kinase pathways and tightly controlled feedback loops. Think about how the active ATM kinase acts in the cell cycle regulation pathway. It constantly scans, and the moment it recognizes double-stranded DNA breaks, boom, ATM kinase activates and triggers a whole cascade. It phosphorylates downstream targets, which eventually inactivate key complexes. The end result? The entire cell cycle halts. This pathway literally acts as our molecular brakes, protecting us from fatal diseases like cancer. So before we wrap up today's explainer, I want you to ask yourself, if a tiny single 105-degree angle in water drives all of biology, and microscopic ATM kinases can completely halt an entire cell cycle on their own, how do these tiny, microscopic thermodynamic rules seamlessly dictate our full-body homeostasis? It's mind-blowing, right? That is the true beauty of biochemistry. Think about that, keep visualizing these pathways, and we'll see you in the next high-yield deep dive. Keep revising, stay focused, and all the best.